on the PCR reaction. Yesterday, we could not have that presentation because Dr. Ranjana from IITA was traveling, and then we could not have that presentation. So today we have the presentation on PCR reaction, and then this presentation from Dr. Ranjana, who is at IITA, will be followed by two other presentations from Dr. Zakaria Kehel from ICADA, based in Morocco. And then the last presentation will be from Carolina, who is a scientist at CIMIT. So without further delay, I will give the floor to Dr. Ranjana for her presentation. Thank you, Ranjana. You have the floor. Thank you.
Thank you, Dr. Mary and Alessandra, and good morning, good afternoon, good evening, wherever you are. Let me just share my presentation, and then, yeah, can you all see my presentation? Is it okay? And I'll put it on presentation mode. Yeah. So I would like all of you to please raise your question in the as Alessandra has said, and this is on polymerase chain reaction, which is also called as PCR. And this PCR is based on certain chemistry or genetics behind it. So I'm just going to give you an overview. Please see it very well because most of my presentation is animated. Yeah. So for better understanding, etc. So to start, the process actually has three steps. One is DNA extraction. We get the DNA from leaves or seeds, etc. And then the PCR amplification. That means we amplify a segment of the DNA. And then we do sequencing of that DNA or many other things that we can do with that PCR product. So the first is DNA extraction. So what we do, these are the cells with DNA inside the nucleus. And then what happens, you do DNA extraction following a lot of steps, and then you get the DNA, which should be at a purified form. It should be completely purified of all the other debris, even RNA, proteins, etc. So what does the cell consist of and what are its components? So the cell is, if you are like this is the cell is made out of 70% of water, and then it will have 30% of chemicals. What are those chemicals? So you will have DNA, which is only 1%. And then you have other like phospholipids, for polysaccharides, RNA, and proteins. So if you are working on DNA, you should make sure that you get rid of all of these things. If you are working on RNA, then you make sure that you get rid of all other things. So you need it in pure form. That's why we need to do quantification and assess the quality of DNA or RNA extracted. So what do we do? It's a cell with all the debris in it that I mentioned. Then what you do is you do cell lysis. You break the cell wall and cell membrane depending upon animal and plant tissues that you are working on. And then what you do is you get rid of RNA by using RNase A, proteins with proteinase K, lipids, etc. with SDS, CTAB. You can get them their full form somewhere. It's a big long presentation, so I'm trying to cut down. And then ions, etc. using EDTA, and then you get the pure form of DNA. So then comes PCR amplification. So what is that? You have the DNA, and here you don't amplify the entire DNA. You amplify a segment of your interest. How do you identify that segment? I will come there, but these are with primers. So then this is your target gene or target portion. Then what you do is you get your target gene, and then you do PCR amplification, and you get multiple copies of this PCR, this particular segment, and this is called your PCR product or amplified target product. So what is the genetics? How did the PCR, was invented and all of that? So we all know about DNA replication. If DNA replication or DNA polymerization did not happen, we will not be what we are. It's because of DNA polymerization that metabolism happens, everything, growth happens, and we become what we are. So what is the building block? The building block is a deoxyribose nucleotide triphosphate, DNTP. So this is the structure where you have a sugar and you have three phosphate groups. And I want you to really look at it very, very well. Three phosphate groups, and then you have a OH here and normal H here in the sugar ring. Okay, so this is to know it very well, and this is all chemistry. And this is the base, and the bases are adenine, thymine, cytosine, sorry, uranine, and cytosine if you are using DNA, and it gets replaced with uracil in RNA. So ATGC, adenine, thymine, guanine, and cytosine. Okay, this is the language of DNA. You know that English language has 26 alphabets, right? And different combination of that will give you a word. While in case of DNA, there are only 4 alphabets, A, T, G, C, and 3 of them come together to give you a word or an amino acid, and that's your building block. So the basics is that this is your existing DNA strand, and that's where I come. You saw 3 phosphates here, but you are now seeing only one here. So why? How does it come? So what happens is, so this is the existing DNA. Then this is your DNTP, right? The DNTP now comes and attaches at the O here, the OH that you have seen, but here you see only one O, sorry, one O. But here you are seeing that OH, again there is one OH, and there are 2 phosphorus. If some of you know what is PCR, we also use MgCl2 in the PCR reaction. So what happens is when this comes and the NTP comes and attach at the O, so then what happens is the 2 P's and the H goes away, and there is a bond that is formed which is called phosphodiester bond. Okay, so these 2 P's and and the H remaining from here they get with MgCl2, and then they form magnesium phosphate plus hydrogen chloride, so that the reaction gets completed, and then this is how the base will come and attach, and the DNA strand will extend. So similarly, you will have another DNTP that comes, and phosphodiester bond is formed, and that's how the DNA extension. This is the basic principle of DNA polymerization. So what you need is, you need a DNA template, you need the primers, which are short oligonucleotides that are already present in our cells. Then you have DNA polymerase. This is a restriction enzyme which is also present in our cell. Then you have the DNTPs, which are also in the cell, and MgCl2, which is also there in our cells. So these are the different components of DNA polymerization that are present in the cell, and this is how DNA replication or polymerization happen. So if this is your DNA strand, because the DNA will, I will come there, but it divides, replication means it first, there are hydrogen bonds between the 2 strands of DNA. So I'm just now showing the one strand, and I'll come there in the next slide. So this is your primer. Primer will always attach from 3' to 5'. This is the orientation, 3' to 5'. So the primer will attach here, and then you have the DNA polymerase, which is an enzyme, and then the DNTPs will come, and that's how your DNA replication will complete. So that's how the double-stranded DNA is formed, and these are hydrogen bonds in between. So what happens? How does this replication happens? In PCR, we use temperature regimes. But here we are using in the cell, how does it happen? Because there are restriction enzymes, some of them are endonucleases and some of them are exonucleases. So we have different enzymes, some of them are called gyrases and helicases. So helicase is an endonuclease which breaks the hydrogen bonds in the DNA strand, and gyrase is a exonuclease which is from outside. It comes and pulls the DNA and makes it apart so that a replication bubble will be formed, which is more like an eye, and then they will keep the gyrase will keep it separated, and then primase will come and attach the primers, and then DNA polymerase will come with the DNTPs. It will complete the replication. So just look at it. So you see, helicase breaks all the hydrogen bonds. Gyrase will keep it pulled. Yeah, I can show it again if you want. So this is what happens. Helicase will break all the hydrogen bonds. Gyrase will just pull it apart and then separate the two strands. And then the primase will come and attach, and you see it's always attached from the 5 prime, 3 prime to 5 prime and 3 prime to 5 prime. So this is what it does, and then the primers will be attached. In this case, the entire DNA gets replicated, while in PCR, only a segment of it gets replicated. So this is what it is in DNA replication. It comes and attaches, and then the DNA polymerase comes, and the DNTPs as well as MgCl2 acts, and then the entire replication is completed. So you have one DNA now you have given, it has given you two strands. So in polymerization, what happens is this is your DNA template. The second is your primer. Then the third is your DNA polymerase. Then your DNTPs. So DNTPs and then MgCl2. So when the DNTP comes, then it attaches on that OH, and then the phosphodiester bond is formed, and then your DNTP gets attached. Likewise, DNA polymerase will move, and then the next DNTP will come and attach, and this is how the DNA extension happens. So now coming to PCR itself. So in the PCR, you do a cycling process, and it has got different steps like temperature, but it uses temperature cycling. So instead of using gyrase, helicase, and primase, we use temperature. While DNA polymerase, we use it naturally found Taq polymerase, Thermus acuticus, which we can find in hot springs, and we can get that, and then we can use this one for PCR. So this is one of the costliest, kind of component of PCR. So then we use also pre-made oligonucleotide DNA primers to use it, and you can order it to many companies like Thermo Fisher, etc., and you can get them. And then DNTPs also you get from companies that produce. So what in PCR we do is denaturation happens at a very, very high temperature, which is 94 degrees Celsius. And Taq polymerase, that is Thermus acuticus, which is a DNA polymerase, it can withstand very high temperature because it is found in hot springs. So that's why we can use it as DNA polymerase. And then your annealing happens. That means the attachment of the primers to the DNA happens at annealing temperature that is called 65. So when you order primers, they will give you the temperature of the primer. But what happens sometimes during shipping, so the temperature may drop, the annealing temperature, and your amplification or PCR may not work. That's why there is another method called touchdown PCR, in which we can actually give a range of temperature, a 10-degree range. Let's say from 45 to 55 or from 55 to 65. So what happens is in a PCR program, you program it in such a way that in every cycle, the temperature will reduce. What does that mean? When the temperature is at 94, the denaturation happens. That means the DNA stands get separated, and then the temperature comes down to, say, for example, if you have used 65 to 55, then it will reduce to 65. If the primer's annealing temperature is 65, it will get annealed. But if it is at, say, 62, then this will continue to reduce every cycle 1 degree Celsius. That is from 65 to 64 to 63 and 62. And that's when the primer will anneal to the DNA strand. So depending upon the annealing temperature, it will attach. This is very good because when there is a change in the temperature of the annealing temperature of the primers, we can actually get this working, right? But the disadvantage is we may not know at which temperature annealing really happened. But that does not matter because as long as we get amplified product, the annealing temperature, at what temperature it annealed, it does not matter, but it really makes it successful. Then there is an extension by using the addition of the DNTPs that happens at 72 degrees Celsius, and it is carried out by DNA polymerase. And these steps are repeated 25 to 30 cycles, and that's how you get a large number of amplified products. So this is a thermocycler program that you can write. So you write like initial denaturation at 94 degrees Celsius, 2 minutes. Then the cycle starts. So if it is a 1330 cycles, then this will be repeated 29 times. So you again do denaturation at 94 degrees Celsius for 30 seconds. So somebody may ask, why do you do 94 and repeat 94 again? This is mainly because the hydrogen bonds are weak bonds. So if you don't do this denaturation, the two strands may come and join again. So it is to ensure that the two DNA strands are separated, and so that the annealing can happen and the PCR product or PCR can run successfully. So this is repeated, and then final extension is done, and then you can keep it at 4 degrees for a long time. So how does this happen? Denaturation happen at 94 degrees Celsius. So what happens at 94 degrees? The both that DNA hydrogen bonds break and the DNA strands are separated. Then comes your annealing temperature. So during annealing temperature, say for example, it is at 65 degrees Celsius, the primers will now come and attach. But the primers are attaching only not from the end, but from where your target is. So again, this is the 3' to 5' and 3' to 5'. That is how it attaches. Then comes your extension, where DNA polymerase at 72 degrees Celsius comes, and the DNTPs also comes, and then the strand is completed and the amplification happens. So if you look at this, if this is your template DNA, and your desired sequence, target sequence of interest is this one, then your you get the primers that will flank it, because that's how the primers are designed to flank the target sequence, and then you will have the target segment amplified containing the region of interest. That's how you get the PCR product. So next few slides are animated, so please look at it very carefully. So if this is in cycle 1, this is your target sequence of interest, and then your DNA, denaturation happens, and then the annealing happens at a certain temperature, and then your extension happens, and then you get this like this. So your DNA copies and target copies, and then you come to cycle 2, and in this cycle 2 again denaturation happens, and then annealing happens, and then extension. Likewise, your DNA copies and target copies are now increasing. Cycle 3, this is how it goes on. It separates, same annealing, and then again extension, and this is how you get your cycle 3. Then cycle 4, I'll go a bit fast. And then you have cycle 5. This is how you are getting your, your amplified products, and you see your copies are increasing. So at the end of cycle 5, imagine how many products you have already got. Now, if you put them in an exponential way, with the first cycle, 10 cycles, look at the number of DNA copies and target copies that you get. They become almost the same. And then after 15 cycle, this is what the number becomes. 30 cycles, I can't even read it, how many copies. And this is how, the number of copies increases, and that's how it is. So I can't even read how many copies it becomes, but your amplified product is up to that level if it's a successful PCR. So what do you do? I just want to go quickly about DNA sequencing, mainly on the Sanger method, but which is based on PCR itself. So what you do is, here again, you have seen the base, right? The three phosphates group OH, H, etc. This is where the DNTP come and attach, right? In case of, when you want to do sequencing analysis, you will see that the O is not here. It's only H. So we call it dideoxyribose nucleotides, nucleotide phosphate. It's the same method, but here we just remove the O and this is only H. So what happens is no more DNTPs can come and attach, and this is called chain termination. So you terminate, there will be no more extension of the DNA. And in this case, sequencing, you don't read both the strands. You read only one strand because you are sequencing that particular strand. Okay, so that's how it is. So this is the model or method that is being used, and the bases like A, T, G, C, they are fluorescently labeled so that when they go through the light, ultraviolet light or infrared light, then these can be detected in a machine. So we use machine to read that. So this is how it is. So this is a normal, this side is the normal extension of DNA, and this one I will show how termination happens. So this is how normally the phosphodiester bond will be formed and the DNA extension will happen. But in this case, look at the right one side here. What happens is now you have a fluorescently labeled base that has come, that is thiamine. The same, because there is a O, the phosphodiester bond will happen and it will attach. But here there is no O. So the next DNTP, when it comes, it cannot find the O, it cannot attach, the phosphodiester bond cannot be formed, and then the chain is terminated. So this is what it is. So what happens is, there's a lot of, there's a same sequence reaction like PCR reaction, but here, in case your DNTPs are fluorescently labeled, but you use all the other components similar as PCR. So it's based on the thermocycler. So denaturation happens, annealing again happens, extension happens, and these are again repeated 25 to 30 cycles. And then there will be labeled DNTPs that we use. So how does the sequencing reaction work? So this is only one strand that you will be now reading. So the primer comes and attaches to the sequence, and then DNA polymerase comes, the DNTPs are coming, and then there is a DNTP that is already labeled. So what happens is, it does not, the chain gets terminated. So what happens is, you keep reading the same strand again and again and again, in different lengths. So what happens is, it continues to read the same, same like this. It will be reading the same strand again and again and again. And then what you do is, you arrange these strands from shortest to the longest. Okay, that's how it will be arranged. And this has, all of this has read the same gDNA strand. Now we use, different different capillary electrophoresis, and there are advanced, kind of sequencing machines now, and there are advanced methodologies. What I'm showing you is the Sanger method, but there are now other methods like genotyping by sequencing, whole genome sequencing, DART sequencing, a lot of others. But the methodology is, so what happens, these are capillary electrophoresis. So the capillaries will carry the DNA, those fragments, let it pass through a detector, and based on the fluorescence it will give you the the reads. So how does it happens? So as the sample travels through the capillaries, shorter fragments will go faster than the longer one. So if this is the light detector and these are your DNTPs, then sorry, those fluorescently labeled DNTPs, so when they pass through the light detector, it keeps reading. You see, it's reading now. A, T, G, and C. So this is how it is read. So this is how you got your sequencing reaction. Then these are arranged, they are put into the capillary electrophoresis. They get arranged based on their size, and then they will be running through the detector, and then they get read. So this is the read of your DNA sequence. So this is how then, if you are actually looking at ABI machines and the results in the computer, this is how your computer read looks. Yellow will be actually read as black, and then this is the sequence you get. So this is the sequence of your own DNA that you have extracted, your own, say, maize DNA. So this is what you have got. Now, what do you make sense out of it? How do you make sense out of it? So what you do is you go to the national, a lot of sequences are being submitted by different researchers in your crop or animal of interest in NCBI, the National Center for Biotechnology Institute. They always submit. So what you do, you take your own DNA sequence and you blast it. Though it's not a bomb blast, but it's a blast called basic local alignment search tool. Yeah, you take your unknown sequence and you match it with already available known sequences published in the gene bank. This is called, gene bank for all the sequences. So you can then, compare those two, and then you can find SNPs, the single nucleotide polymorphisms, that can be used as a marker. You can do gene identification. You can do your organism identification and a lot of other things that you can do. So this is how BLAST is done. And if you want to go and register yourself, it is freely available. I will request the participants that you should go and try this yourself. Yeah, just you don't even need your own sequences, just go there. A lot of sequences will ever be available. Try to do the blast and learn it. So what happens is query 1 is your own sequence, while subject 52 is the already available sequence in the gene bank. So if you look here very carefully, you will see that there will be, I'm trying to find a what is that called, SNP. I didn't do it earlier, so I'm trying to find now. If you see, there is a complementarity. Here is the SNP. You see, here they are all complementary, right? A, G, G, T, T. But if you come here, or am I seeing wrongly? I think they are all same. G, no, this is a C and this is a G. So there should have been a G, right? But instead of a G, now there is a C, and in this one there is a G. So this is a SNP. So likewise, you will go through all of these sequences and then you will find a SNP. So this is how SNPs are identified. You can also do your primer design doing DNA sequencing. You can do a lot of things with your primer sequencing. And to just give you a good, presentation overview, this starts with leaf sampling or blood sampling, depending upon the species you are working. Then you do your grinding. First you lyophilize the DNA, samples. Then you do grinding. Then you do DNA extraction, PCR, gel electrophoresis or capillary electrophoresis, then you do DNA sequencing, and then it is all about bioinformatics. So in a nutshell, what I'm saying is that the PCR is based on certain genetics, which is based on our DNA replication, and then the DNA sequencing is based on chain termination, etc. Thank you very much.
Thank you very much, Ranjana, for this very nice presentation. So, dear participants, with this presentation now, we have completed the presentation on where we try to establish the molecular biology fundamental. So now we will see from the presentation from Dr. Zakaria and after Dr. Carolina, we will see how all these techniques have been applied in your gene bank collection to generate data, to know the extent of diversity in your collection, to create subset. So let us now, without further delay, give the floor to Dr. Zakaria for his presentation. Thank you. Zach, the floor is yours.
Do you hear me? Or...
Yes, we can hear you.
Okay. I don't need this. Good morning. I will share my screen. Okay. My screen. Please let me know if you see my screen now.
Yes, if you can put it in presentation.
Yes, I'm trying.
Perfect.
Is it okay or it's
now.
coming weird?
No, it's perfect. Thank you.
Okay. So good morning, everyone. Yeah, thank you for being here. As Marie-Noël said, we will spend almost 1 hour looking at how we use all this information that we are generating from genomic characterization, markers, etc. I'm just putting here, of course, this is my email in case this is not only my work or the work of Ricarda, it's coming from the whole team. The way we are tackling this issue of using genomic in plant genetic resources, but also in the use for breeding, discovery, etc. Here are some examples coming out from big collections on using high-density genomic information of characterizing collections for different objectives that I will be touching upon. So what I want to do here, I know, while we are working with the digital sequence information, we saw a lot of tools coming out, a lot of data. The game has changed because before we have little small amount of data that we can handle in Excel. We can look at it. We can see all the patterns. With the genomic and sequencing, it's very, very hard. And we see a fashion of people taking those big data sets, analyzing them without looking actually at some details that are very important. And hence the title of my presentation, including data cleaning and interpretation, etc. So yeah, looking at the cleaning of the data. Again, a lot of this data, we cannot see it. We cannot just scroll it in Excel because it's big. Most of it, we cannot even open it in Excel. We just take data from different format and we use it in a software. We get a couple of graphs. We are happy with that. A lot of colors, and we start interpreting. However, I'm trying to give some real examples to show how this can be dangerous actually in terms of analyzing, using the data, interpreting it, and also the way we use it in our routine activities in conservation, for example, in gene bank or use in terms of using diversity. You saw the presentation just before, and I think you had a lot of presentation on how we generate actually sequencing information. That comes with a lot of errors, including sequencing, missing values, contaminations that we can have, variant calling. We can have false positive and false negative. And this is important because if we don't deal with all those issues, we will have a problem with statistical power or analytic power. We will have non-accurate comparison because we are trying to compare, we are trying to identify genes, as has been said before, and then we may end up having a false positive gene. We if we clean well the data and look at the quality of the data, we will obviously have less issues in downstream analysis like genomic selection, genome-wide association studies, diversity analysis that we are using. So I'm going with you just here to show, first, we saw a lot of paper what is a really difference between diversity and structure. So diversity, we want to see how much diversity and how much genetic variation we have within and among individuals or populations. And that's, we have indicators. The indicators are allelic richness, number of alleles, expected heterozygosity, or what we call genetic diversity, polymorphism, genetic distance between individuals to see which one are closer to other ones. And we use that mostly to understand, of course, the diversity in a collection, but also assess the completeness of a collection, compare it to another collection, see if we have enough diversity or not. Redundancy between collections, I will be giving some examples. Provide maybe and looking for new variation that it's not present and needed by breeding program, looking at the fact that breeding program don't need all the diversity, but useful diversity that we can use genomic information to identify very well that new diversity. And also how we use all this to maintain a healthy genetic variation within a breeding program. And that's, for example, can be an example of SNPs diversity across a wheat panel to identify accessions with novel alleles for salinity tolerance not existing in a breeding panel. That's something completely different from the population structure, where we actually both are useful but for different objectives, where we are looking at a non-random distribution of genetic variation. We look at subpopulation with distinct allele frequencies. We look at stratification by different, parameters like geography, climate, breeding history, domestication event, social events, sometimes, or nutritional quality. We can find that. And how we do that, we use, of course, and that's why I'm coming, because we are looking at the analysis. We use all, groups of analysis, including structure, which is based in clustering. Again, those tools, they give you nice graphs that you can interpret very well, but we need to be careful what actually happening inside the tool. Very simple analysis like principal component analysis, phylogenetic trees, etc. This is very important, and we will stop later in a slide or two, because very, very important to correct for false positives in association studies and QTL mapping, understanding also the adaptation of subgroup to specific environment. I think we are doing a lot of work, different centers differently. I will give an example on that, but also that helps you also looking at the structure that you can use to design crosses that will be useful to maximize a combination between structured group. For example, if I look just an example of rice, we have very clear population structure between Indica and Japonica, for example, where we have clear allele distribution and patterns across the two groups. This is in summary how we use it. They actually, they are complementary, but they are not necessarily the same thing. It's not the same definition. It's not the same question. It's not the same focus. Why we have diversity versus why we have population structure, the importance for breeding programs, but also if I give you an analogy to see, it's like if you are comparing diversity of a library to see how many books, for example, as an indicator we have in a library, so we can compare two libraries. How many languages are present in books in a library, so you can compare. That's an indicator, like if we are looking at allele richness compared to population structure, where in a library we look actually how books are organized in shelves. Is it by author, by team, by language, by age? And that's completely different objectives. As I said earlier, they are complementary, but they are not the same. One thing that I want to touch upon here is really the way we analyze. We have been looking at analyzing DSI, and we saw a lot of papers there. The availability of free software has made it very, very easy for people to analyze, and unfortunately, a lot of understanding is missing on the way why we use one on the others. Sometimes we use both of them. Again, multivariate analysis, like principal component analysis, coordinate analysis, they serve something specific, which is a really reduced high dimensional question we have in genomic information to few axes that may show you kind of gradient of genetic variation. So you plot them and you see if you have some clusters. We will look at an example later compared to Bayesian analysis or structure analysis or whatever similar methods where actually you need the prior information, like the example before of rice, indica and japonica, where actually you say, well, in my rice collection, I have two subpopulation. Let's see if the data will show that or not. There are limitations in terms of using one of the other one, but again, they are complementary, and we need to really see what's the objective of the analysis to go further and analyze the data and be able to interpret so we fit our objective from the analysis. So now I will, before going to some examples, I will take some time to explain this because we saw we work a lot within centers with partners, a lot of students, and we know how fashionable the use of GWAS, trying to find an association between a chromosomal region, a region in the chromosome, and the variation in a phenotypic observation of a trait. So it's just see if that variation is linked, the variation we see in the field or the lab, is it linked to a variation we see at the genome level? And most of the time, I'm coming to the one that we are using mainly in genome-wide association study, which are, for example, mixed model. And I will show you why we are using that. This is an example where we actually try, I don't know if you see my pointer, where we have a marker effect that we want to see if it's significant for that phenotypic variation or not. We have some population structure that we need to account for, and I will show why that's needed in the next slide. And of course, we have some random variation coming from the individuals. We account for the kinship matrix, which is a relatedness matrix that will give you an idea how individuals are related, because that will reduce the amount of false positives or false associations that we will find by model. And here, I will give, of course, more details on that because that's part of the cleaning we are doing. And of course, we need to make sure that our residuals or errors that we cannot explain by the genetics are quite controlled, etc. So if we look at this, if I take an example of a panel of 1,000 line in a breeding program or gene bank collection, and I have, let's say, just 10,000 markers, this model will need to run 10,000 times. I mean, 10,000 times for each marker, you will need to run this model. And every time you run this model, mathematically inside, you will need to inverse somehow the kinship matrix to be able to solve this equation. And the matrix has a size, for example, of 1,000 by 1,000, because that's the number of individuals I have in my sample. If we give just 2 seconds per model to be run, that means we are talking about 10,000 by 2, which is 20,000 seconds to run this model. So a lot of research has happened to reduce the time, mainly in animal, human, and pharmaceutical, and we did a lot of approximation, etc. So what happened, and I'm saying that because we have seen in papers that were reviewed that we are using a lot of approximations and compressions that we don't understand very well, and they're affecting our analysis. If you look at the example I gave before, 10,000 marker and 1,000 individuals, taking 20,000 seconds, that's a lot of time, people do compression. And compressions mean that rather than using a kinship matrix of 1,000 by 1,000, what we do a clustering analysis using genomic information, and rather than using a kinship matrix of between individuals 1,000 by 1,000, we will find 10 clusters, and then we will be able to use a matrix of 10,000 by 10,000, assuming that this is giving us all the variation we have in our panel, which is reducing the time, but a huge approximation on actually affecting the false positive we will be going to and to identify. The other approximation we are doing is what we call population parameters determined. When we go back to the model, the initial model we are using in association studies, rather than going and solving this equation 10,000 times, and every time we need to have a solution. Solution will be a p-value for a marker. It will be heritability and unknown genetic structure, but also the error variance I'm putting there. So every time the model is running, and that's why it's taking 2 seconds, because it's actually solving this equation and giving you the results or the solutions of those at least 3 parameters. So when we do compression or population parameters previously determined, or what we call P3D, that will run the model one time, will fix those parameters, and every time you run a marker, you just use the previously recorded parameters, which is a huge, again, approximation, and will affect, obviously, very hard your outcomes and marker associated with your variation in the tree. Why I'm telling you that? I'm telling you that just to tell the colleagues that this is part of looking at cleaning the data, but also looking at the models we are using. We should not just blindly use a software because it's freely available, but rather look at what the software is doing to be able actually efficiently to interpret the results. Let's go back to the structure, and we said structure and diversity. I have a lot of use cases that I will show, not a lot, don't worry, that we are using in Genebank and breeding. But we are also using structure and diversity to look at the population effects, for example, when we are running genome-wide association studies. And I will give you here an example just to show why it's very important. Let's say we are trying to find associations of a panel with plant height, wheat accessions, and those wheat accessions, I have half land races and half breeding material. And I know very well that some SNPs alleles are more common in land races. If we run a GWAS without correcting for population structure, and that SNP may be appearing that it's associated with plant height, but that's not true, not because it has a causation or it's controlling plant height, but simply because it's rich in the land races, looking at land races having higher plant than breeding material, that they are shorter. So if you don't do that, we will run a genome-wide association study, and we will then identify that SNP that actually is false positive. It's more linked to population structure to actually controlling the plant height itself. And again, we use a lot the kinship matrix, and the kinship matrix actually give an idea on the relatedness. We call it also in the association. And I will give you an example in a case study where we are trying to find associations with grain yield. And we are using a panel only with wheat breeding. But however, we have a lot of blocks of sister lines coming from the same breeding program and sharing a long part of the genome. Those lines also, for some reason, are performing well and giving a good grain yield. If we don't take into consideration this relatedness, we will just pick up false SNPs that they are more, affecting or coming from the ancestry rather than the causality with the grain yield, in the same way we saw for population structure. So this is very important, and that's why the diversity and population structure, we need really to study it very well once we clean the data, looking at the software, etc., to be able to have outcomes that they are clear from false positives or false negatives. Another aspect that we realized as we analyze, hand density genomic information is really looking at the kinship. We saw that there are little evidence of people cleaning the kinship matrix, and because we just do a cleaning a kinship matrix. Kinship matrix look at it as a distance, a relatedness, genomic relationship matrix. How individuals are linked together using genomic information. And this is very important because we use it extensively in, of course, understanding the diversity, but also in terms of correcting for relatedness in genome-wide association studies and genomic selection. If we don't reduce the noise in kinship matrix, we will inflate the result and we will have false positive. And why we are having this problem, you will tell me, if I take a matrix of traits and I get a correlation matrix between traits, so I will not have this problem. Yes, we will not have this problem because it's not that high dimensional. For kinship matrix, especially looking at all the errors in call rates, but also missing data, we see some strange patterns. And unfortunately, a lot of users don't actually diagnostic all the problems we have in kinship matrix. As an example, I will give you an example, the diagonal. If I look at the diagonal in kinship matrix, this is the relationship between an accession and itself. And depending on the methodology the user is using to compute the kinship matrix, that should be, 1 or 2, right? The off diagonal is really the relationship between two different accessions, and in general, it should be more, less than 1 or less than 2, and they will be more centered around 0. However, due to, sometimes missing data amputation, we see some weird pattern. For example, diagonal less than diagonal should be higher than off diagonal, of course, because an accession is more close to itself than others. But sometimes, unfortunately, we see different patterns, and that's something that one needs obviously to look seriously before going for further analysis, because that means there is a problem of the data, and that needs to be cleaned before going further in genomic selection or GWAS analysis. We need also to look at duplicates because the kinship highly or closely individuals will obviously affect the results. That means they have almost 100% similarity genomically, but then variation in trait, and that will affect the power of any analysis we are doing using kinship, like association studies or genomic selection. We need also to look, because as I said in my first model, somehow when we run GWAS or GS, we need to inverse a matrix. However, that matrix, unlike the one that we are using normally with normal traits, it's not necessarily symmetrical, and it's not necessarily positive definite. That means we cannot inverse it. And if we cannot inverse it, we do a lot of approximation and pushing it to be able to inverse it and solve the equation. So all this needs to be there. I know it's a heavy stuff, but it needs to be there before going and just pushing this in any software and getting some analysis that, look great, but behind actually they are full of inflations and full of false positive. Let me now share with you some use cases that we are using and how we are using those methodologies in GeneBank and beyond. This is just an example of diversity analysis using principal component analysis. It's group of land races from a collection of barley, and you can see that the PCA analysis as an initial analysis will give you an idea how many groups you have. If I look here, I may see I have 2 groups. One of them is very distinct in the axis of PC1, which will give me mainly a separation between a group from Ethiopia and another group having China and Turkey. But then maybe I have another differentiation in the second axis, PC2, where, you know, China subpopulation actually having lower coordinates. So now I start looking and I say, okay, my collection, I may have 2 or 3. Let's go beyond that and see how it looks in reality. So we go and look seriously and more starification in and using some other tools that they are more powerful than just statistical analysis. And then I know that I have 2 or 3, so I can start testing. And I say, well, yeah, I have, you know, let's consider that I have 2, and this is what I have in the first part of the graph. And that one, you know, I can see that still, yeah, I have Turkey and China still there. And Ethiopia, for example, it's completely different group. I push it back and I say, okay, let's try maybe 3 subpopulation. And here, suddenly, I start seeing that actually, you know, there is a separation between China and Ethiopia, and that's give me more logical sense because, you know, the eye of the expert or the curator or the molecular biologist or the reader is very important to help interpreting this. And then I say, okay, should I stop or go beyond that? No, I will try, what if I have 4 subpopulation? Then I start seeing some noise, like, yeah, not that clear, really. So I can stop there and say, well, this is enough for me. I have 3 subpopulation. I have an explanation. They are based on original, actually the origin of the collection, making sense. I go forward. I may test this by using, you know, analysis of molecular variance and see which group is giving me more variability or more distinction between groups. Is it number of subpopulation 2 or number of subpopulation 3? And then I will take the one that is actually maximizing the differentiation between the two, and then everything is okay. I go with that one. So this is very, very important. It's 3 categories. Running a quick PCA to get an idea if we can see something, then go to more in-depth analysis, and then run some diagnostic to see if really my explanation of having K equal to 3 as subpopulation is the one that I need, and I don't need to go to K equal to 4, because if I take 3 rather than 4, that will affect my analysis later in, as I showed in genome-wide association studies or genomic selection. So that's very important. And then I can start looking at diversity, and that's what I will show how we are using it, look at the diversity within group, etc. One thing that I want to mention here is that all depend of the sample we are using. In this example, I'm using subpopulations from China, Turkey, and Ethiopia. I do data cleaning, I look at the kinship matrix, I start analyzing PCA and genetic structure or population structure. However, if I add, let's say, population from Mexico and another one from Morocco and another one from India, the results may be different. Maybe Turkey will not be with China anymore, and it will be more closer to Afghanistan or something different. So everything is relative to the panel of markers you are using and the panel of individuals you are using. So similarities and dissimilarities may change depending on the panel used. And of course, we can look at which one, if it's making sense. This is a genetic distance between the subgroup. And we can see, of course, that China and Turkey are more genetically close than to Ethiopia, which is something that we saw already here in the PCA analysis. I mean, in any analysis we are doing, simple analyses are the best. If I run PCA and it gives me all the outcomes that I want and they are making sense, I don't need to go and do complicated analysis that I don't understand, or Bayesian models or some complicated machine learning models that they will give me maybe better graphs, but they will end up making just a lot of inflation of the outcomes and making some analysis that I will be, as a biologist, unable to interpret. So simple is always the best. If simple, I want to validate it, I go further and then I interpret as I go. Another important aspect that we are using, especially in Genebank, is what we call spatial genetic structure. Spatial genetic structure is beyond just population structure, is looking how space, when we call space, it's geography, it can be climate, environment, soil, sociological aspect, how they're driving actually the adaptation of cultivated species mainly. I'm not talking about wild species in the case of GM. And we have two categories in looking at spatial genetic structure or spatial structure. We have one, what we call explicit genetic structure, where we run PCA or we run Bayesian like structure software, and then we map them using our passport information and see if we have a pattern or not. And that's something that we can see here. We have very clear differentiation between three subpopulations. We have a very distinct group in Ethiopia here. We have one which is admixture between two, one in Turkey, one in China, but we still see that, the genomic makeup of Chinese subpopulation, it's quite different from the one of Turkey. So that give us an idea why some distribution of alleles is more in a region or not, and we can correlate it with sociology, climatology, soil, and other parameters. But we can also be implicit. Implicit, I mean that you use our passport information that we have in GeneBank because we know where we collect the information and go beyond and see, do we have something that we can actually use to differentiate between population and effectiveness? And here, we use some tools that they combine actually geography and molecular markers, both together. It's not mapping in a map outcomes from structure or diversity analysis, actually using them together. And here we are looking at two components. One of them is local and global genetic structure. This is very important for us in GeneBank and the understanding of distribution of a collection. Global diversity or global structure will give you differentiation between groups as you go into geography, between south and north, between east and west, etc. And that's very, very important because it gives you the effectiveness of a SNP or marker to reveal large or important pattern and also structure between individuals or population. And I can say my subpopulation in northern Morocco is completely different genetically from the one in southern Morocco. So that's good. The other one that this kind of analysis will give us is really the ability of a SNP or marker to actually discriminate between local land races. I mean, if I go to a small region within northern Morocco, I have some local diversity there, and I know that even neighboring accessions, they are not necessarily fully similar genetically. And that's what we call the local structure that will give me an idea on local adaptation of the collection, and that I can use later for conservation, but also for use or to identify gaps in the collection, etc. And that's how we use it. We use it sometimes to identify really genomic barriers that we have geographically that we can identify for our collection. This is a very real example from Doromuit in Morocco, where we can see that there is a very clear separation on genetic makeup between the one that they are in the high Atlas mountain and the mountain in the northern of Morocco and the others. That means if we are looking for something interesting that it's in the subpopulation coming from high altitude, we know where we find it because we always find it only there. And those kind of spatial population structure that we are doing can help us not only understand that, but go look for new diversity if it's needed. I will go almost to little bit summarize and see how in GeneBank initiative across the CG and others also working with some national program, we are using this in both because we used to conserve better, but to use better as well. So in terms of conservation, we use this kind of analysis, of course, of digital sequence information to identify duplicates. Identify genetically close accessions that help us in GeneBank, for example, to have a clear plan for what we call dynamic management of the collection, where GeneBank is focusing more on a portion for now because that's useful than focusing on everything until you see use for the other one. We look at, like at the example here, misclassified land races or species. We have, sometimes we have that. You go and you collect something, and then the passport that tell you it's this or this, it's winter or it's spring growth habit or it's this species. you put it in the field, it's not always easy, but the marker will give you that, yeah, those ones actually are misclassified. So we can put them in the field, verify again, and then correct our passport information, because that information we are using in Genebank to be able to get to users with the right material that they need for their use cases. But also we are using it in genetic structuring that's not easy always using passport information, that we are using for gaps and identifying gaps in the collection, new variation that we go collect with our national partners. In terms of use, there are plenty of use cases, but I can tell you that we, as we do for marker assisted selection, we can use actually some, and I will give an example later, some associations to actually increase the efficiency to identify traits that they are important for users. We link traits to passport information, and we use genomic prediction to reduce the phenotypic effort that is quite expensive for Genebanks looking at the size of the collection. We use it as I showed before, but examples are coming in terms of marker trait associations, GWAS, or adaptations mechanisms through identification of marker environment associations. Here is again a real example from our colleagues in CIAT, where actually looking at enhancement, understanding of a collection, looking at the variable nature of an accession in Genebank, where genomics has helped identify subgroups within a landrace. If we look at the landrace, and landrace by nature is a population, very variable, but here genomics can help well curators to identify really subgroups, because it makes a huge difference between, looking at the original landrace, which is a mixture of two groups, and the two others, because that will make a huge difference for the users if you send the original one or you send one of the other ones, because they are almost, more pure than the original. Another example from, our colleagues from CIAT and IITA, where you use environmental GWAS, where Genebanks are trying to link in the same way we are trying to find associations between variations in the phenotype and variation in the chromosome. We are trying to find variation in the original climate where we collect material in the Genebank, and the variation in the field. And you can see that we can find absolutely amazing associations that can help us. Let's say in this example, if I look at this one here in the right, this box plot, a distribution of temperature from where we collected a group of landraces that they present only the allele GG or GA heterozygous, or the other one AA. I know for sure that most of the land races that I'm collecting from high temperature are coming actually having AA, and that will help me, if that association is significant, very easily select the material from Genebank based on the presence of allele AA, etc. This is again the same thing. This is another example of association outcome for environmental GWAS on barley, and you can see that there's some strong association that we can use later in the same chromosome, that we can use later to identify. This is a very clear association with I think the decrease of rainfall. And for somebody working in Genebank like me, having that strong association will make my life easy if somebody is asking for Genebank material coming from low rainfall. Of course, we have passport information. We can go and extract climatic data and take the material that we are doing. We have developed a lot of subsetting tools and focus identification germplasm strategy to identify those coming from low rainfall. But sometimes passport information is not valid or it's not available. Markers will tell me that, yeah, I may go focus on the one that they have those alleles that they are actually linked to the reduce of precipitation from the origin. My last example is really the work we did for 22 crops for gap analysis, and you see the gap analysis, especially for wild species, it's very straightforward because we look at the distribution model of a species, looking at how the natural adaptation has, shaped the hotspots for wild. For cultivated, we have worked in Genebank initiative last 5 years to develop new methodology because we look at the cultivated species. We have two parameters. One of them is the natural selection, the way we see in wild species, but the second one is really linked to tradition, nutrition, all this aspect, which is the farmer, shaping the genetic profile and the distribution of cultivated species. So we use in general some categorization of cultivated species. If this example, we can look at the example of bean, very, very clear population structure, which is Mesoamerican and Andean. Again, passport information sometimes is missing, sometimes it's low quality. So what we do, we can identify beyond the race and go further and look at subgroups using genetic structure and population structure. And then we have very clear, distinct subgroups beyond just the races. And sometimes we have structure within a race. And we use that to identify clearly which gap for which genetic subgroup, which is much more, precise than going at the race, because we are going at the level of the genome and the distribution of the allele within the subgroup. And that's much more helpful in case we want to go and target very new diversity that we don't have in Genebank or in breeding program that we can go collect with our national partner. So I know my colleague, I think Carolina will come later. I will stop here. We have some time. I thank you all for listening to me, and I'm happy to answer any question if you may have at this stage. Thank you very much.
Thank you, Zach, for this wonderful presentation. Thank you so much. A lot of information have been shared. So, Alexandra,
I need
please.
to say, sorry, can I say something I forgot?
Yeah,
Because
go ahead, please.
lot of those analyses just for the participant, they will be available in HTML together with sample files and scripts that we will share with the material and at the end of this webinar series. So we'll share with them sample analysis and files for cleaning the data, for kinship cleaning, for diversity, for structure, for GWAS, for genomic selection. All that will be shared by Alexandra and will be made available for participants. Thank you.
Thank you, Zach, for the useful information. So, Carolina, I don't know if Carolina is there. Alexandra, can you help me?
I don't see Carolina yet. I've been trying to contact her. I think it's just very early for her, so maybe she's having trouble joining us. Perhaps we could answer the question that is in the chat in the meantime.
Exactly.
So
I don't know even
who
start the question answer session,
yeah.
And then when she comes, we just give her the floor. Thank you. You have the floor.
Yeah. So there is a question.
Alessandra, is it the question and answer for both me and Zach?
Yes.
Okay, great. Thanks.
So I don't know actually which presentation this refers to. Perhaps you can help me. It says, kinship matrix is possible to calculate for polyploid species or only for 2N species? Is it maybe for you, Ranjana?
No, this is for Zach.
No, okay. This is for me, but I don't have the answer, unfortunately. I think I don't. I know we are struggling with polyploid. We try to do a lot of approximations while we work. My only example was with a little bit, I think, cactus. And it's not always easy. We don't have, most of the models we are using are actually for diploids. So yeah, I don't have very clear answer, but there are models that are approximating, the model to be able to approximate the kinship matrix as possible, as much as possible for polyploid.
Perhaps Ranjana, you want to add something?
Yeah, so it is correct that polyploid species, they are difficult. First of all, we need to know the ploidy status of all those genotypes that we are working with. Majority of the times, we don't know their ploidy status. However, tomorrow next week there is a session on the polyploids, and there will be, people who will be presenting on how to do genotyping and how to do address some of these issues. Probably you should park this question for next week. I'm sure there are people who will answer to that. And I know that in YAMS, we are working with some kinship matrix, but I don't want to answer for the scientist. So let us have that question addressed next week. Thank you.
Thank you, Ranjana. So there is another question for Zach. I don't know, Zach, if you have access to the chat, but the question says, well, it's more of a, I guess,
I have
a
access,
note.
but I don't see it.
So it
No, I
says
can see it. I can see it. I can.
Yeah. I think it's more of a statement rather, but it's important to evaluate genetic structure to correct for it. But also
Yes.
it's important to look for additional information to enrich the analysis. Do you want to add something to that?
No, I think I think Norma has summarized everything very well. What I want to say here, because especially for our student, etc., you know, put in some data that you cannot open in Excel because we are used to use Excel, putting it in a software, getting a nice graph. We need to be careful with that because that has a lot of implication. We need to go step by step, understanding the science behind, and then the software is just a tool. So software is not the most important, but now lately, since we cannot manage, efficiently the way we manage data as genomic information because it's big, so it's easy just to put it in a black box and get a couple of graphs, and that's becoming dangerous because we see that as we review papers and look at the work we do sometime with different partners and students, etc. So that's really exactly what Norma summarized as well.
Great. Thanks so much for that intervention. I will first address the hand up and then I'll move on to the other questions. So we have Dr. Ouedraogo from Burkina Faso. I see you have your hand up. So feel free to speak. You have the mic.
Okay, thank you. You hear me? So I have a
Yes.
question to Zakaria.
Yes.
Yes, please.
Yes, please go ahead.
I think we lost you. Yeah, I think maybe there's some audio issue. So maybe we can come back to Dr. Ouedraogo.
Yes?
Yeah. Hello, can you hear us?
Yeah.
Okay,
Yeah,
we
hear
lost you
me?
for a second. Do you want to repeat the question?
My question is how to define the core collection. Core collection. You understand?
Yes.
Yeah, core collection of accession.
Yeah. Okay. So yeah, core collection of accession is just for explain. Core collection is a portion of the collection, most probably 10% of the size of the collection that's having almost 90% or 80% of the diversity. And for that, we can use different parameters. We can use passport data, we can use trade data, or characterization and evaluation data. We can use marker data. So what we do in general is just or you can combine three of them, right? You use the same way we are doing with the climate is you use a model and starification because it's very important. You need to look at the structure you have in your collection. Why I'm doing that? Let's say you have a collection of 1,000 accession. You need to look at the first at the population structure you have, because if you have three different subgroups and they have different sizes, you need in your core collection to respect that distribution. If I have 70% subpopulation A, 20% B, and 10% A in the core collection, I need to have that. So that's one thing that we do first. And then we use all the genetic variation in a cluster or multivariate analysis to maximize, to have a set of accessions that they respect the stratification, but also maximize the allele richness that we have in the data, at least 80%. So that's how we do it. Again, you can use different data separately, or you can combine, phenotypic, climate, and genomic as well.
Okay, thank you.
Thanks so much for that answer and for the question. Feel free to raise your hands as well. You don't only have the option of the chat. So there is another question by Marta de Grasa. For diploid species such as cowpea, which keys, aspects should we focus on while analyzing kinship?
Well, analyzing, I was talking mainly on diagnostic, because what happened is that we look at the data. For example, in diploid, we have a couple of parameters we check. Do I have just 2 alleles or 3? Sometimes we have data coming from the lab for diploid having 3 alleles, which is impossible for diploid. And so there are some control point that we do. I saw little, first because we don't have tools, and second, I saw little of people actually diagnostic the kinship matrix. And again, kinship matrix, if we look at it as a correlation matrix, it's quite easy. I need to have 1 in the diagonal and less than 1 in the off diagonal. However, the way we calculate correlation matrix statistically is different from the way we calculate kinship matrix. Plus the fact that kinship matrix originated from the marker data, where actually we have a lot of missing data, and sometimes we do imputation. So it's a little bit more messy than just normal correlation. That's why I said it's good to look at the diagonal, off diagonal. Diagonal should be higher than off diagonal. Off diagonal should be centered around 0, because there is less relatedness in off diagonal compared to the diagonal. But also looking at if the matrix can be inversed or not. We have parameters that we control to see if the matrix can be inversed or not. Because if you don't do that, the software as a black box will go and actually approximate your kinship, and having the most inversible matrix close to your kinship, which is sometimes actually quite far from your matrix. So we will be able to give you some codes and examples with the material at the end of this webinar. I hope that answered the question. Thanks.
Thank you, Zach, and thank you, Marta. I hope that answers your question. Feel free to add more in the chat if you need. Then I have a question from Ali Shehade. How can we reduce unspecific bands for the amplified DNA, and which markers are useful for DSI?
Yeah, that's a good question. This all depends upon, firstly, the primers that you are selecting for, the region that you want to amplify. So this actually now depends on, the trait that you are looking for, and if you really know exactly the position of the QTL, etc., within the DNA. So if you know that, then you can generate the primers flanking that region, and then you can get, these unspecific what is that called, amplification of DNA can be reduced. Yes, it's a problem. That is one. The second is, if your DNA is not cleaned enough, and when you are taking your samples right from sampling, if you took leaf samples and there were some fungus in it, or there were some soils in it, so you are going to actually extract the DNA from the microbes as well. So when you do DNA analysis, maybe, or DNA extraction, maybe the DNA from other microbes also came into it, right? So DNA is made out of ATGC, so the primers are also ATGC. So if there is some complementarity, then you will also have unspecific bands of amplified DNA. Which markers are useful for DSI? Depends upon the money that you have in your pocket, right? So if you ask me, I will say, or even Zach, for good quality data, whole genome sequencing. You should do whole genome sequencing as much as possible, or resequencing, right? But these are costly, right? So that's why a lot of people are nowadays doing CASP. CASP SNPs, just limited number of markers that are very specific for identification. So in case of CGIAR institutes, we use medium density DART tag or low density CASP SNPs. Depends upon what you want, right? And for me, doing a genetic diversity and population structure is always a question, because if you have not covered the entire genome, how do you say one is no different from the other, right? Or one is identical to the other? But we do with what is available, and that's why we use stringent bioinformatic analysis, right? And that's why in marker assisted selection, a lot of time when we do biparental crossing, the QTLs are not, most of the QTLs are ghost QTLs, right? We can't even identify or validate them in an additional biparental population. So yes, there are some questions, but if you like precision genotyping, then you need to start with precision phenotyping. Is your phenotyping good enough, right? If you are scoring a disease resistance as 1, what is the other person scoring? Is it really 1, or it is 2, or it is 3? So these precision phenotyping, as long as we cannot do, genotyping, even with whole genome sequencing, you can't have precise position of your traits or trait mapping. Thank you. Yes, AI can be implemented. That's why people are moving to, machine learning, right? We are using more and more image analysis, more and more drones, and more and more mobile-based apps, right, for disease scoring, right? So that's why all those AI or precision phenotyping or high throughput phenotyping is becoming more and more important, because as long as we don't do good phenotyping, we can't have our traits mapped. Thank you.
Thank you, Ranjana, for that extensive, not expensive, answer, and for combining questions. You're right. Indeed, the second question was also relevant to this conversation. Zach, there is a question on GWAS analysis from our colleague at Africa Rice, Arnaud. It is often recommended to apply filters on the minor allele frequency of variants, for examples in the thresholds of 1, 5, and 10. What impact does this filtering have on the results?
Well, yeah, well, that's a very interesting question, because it all depends of which panel you are using. For example, the threshold for minor allele frequency will obviously change between if I'm using advanced breeding material, crosses, gene bank material, etc. So that will have an effect, because the problem of minor allele frequency, depending on your population, you don't know if it's actually a rare allele or just an error from genotyping. So if I'm looking at stable lines from breeding program like generation 7 or 8, maybe I will go even to minor allele frequency of 10%. If I'm using gene bank material and I'm quite happy with the quality of genotyping services and the reduced error there, I may go to 1%, because I don't want to miss a rare allele. So yeah, it depends of the panel and you are using and your experience. The amputation, of course, it has an effect, huge effect, depending. There are several methodologies. You need always to use the amputation that will allow you to do cross-validation. What do I mean? So there are some models that they will just go and do amputation, which is mean or average, moving average or something. You need to look at amputation that they're quite advanced, sometimes that they're using more machine learning, but then they validate the results doing some cross-validation with the known inampute data. So that's the only way of controlling if the imputation model is good or not. The QQ plot in GWAS is very important because that will give you an idea of the quality of the analysis, because that's the not the correlation, the relationship between what you expect and what you get. And that's where we see actually the false positives. So it's a very, very important one. Looking at, so before it was easy, it has a normal shape that we know, so it was easy for people to interpret it. Now with the high density genotyping, it's not that easy. And why? Because data is not filtered very well. Sometimes I was reviewing a paper where we saw some very strange QQ plot, and we realized that actually people were mixing between identifiers for traits and genomic, which give you a strange profile. So if you have strange profile in QQ-plot at a certain level, fine. But if you have something completely strange, you may need to look at your kinship, the quality of your data, your identifiers. I know a lot of problems happening because people don't respect the fact to have the same unique identifier between what we genotype and what we phenotype, and you end up sometimes having files coming from the field using an identifier, which is different from the one coming from the lab, and then you mix that, but then actually you are putting the marker data with the trait data, but for different line, which obviously will destroy your analysis and give you different patterns. So yeah, those are the problem, but the QQ plot obviously should have a normal distribution the way we want. I will go directly to the next one, GWAS analysis for how many genotype appropriate since different research. Yeah, so the panel for GWAS will depend of what you want. Of course, if you ask me, you need a good diversity. It's not only about the size. Obviously, I wouldn't do GWAS with 100 line or 200. I will need more than that. But in the same time, if I have very related material of 1,000 breeding line, very much related, it doesn't make sense to do random GWAS. So it's a combination of a size to be able to have enough power for your analysis, but also the diversity you have. Not too much diversity, but some diversity. Yeah, thanks.
Thank you, Zach. I saw you went through all the points of that question, so thank you so much. I have a question for Ranjana in the Q&A, actually. Can BLAST be used for cultivar identification, or it can only identify species?
No, you can do even cultivar identification if you are using the same, so what you need to do is, in the gene bank, you have sequences that has been deposited. So if you are working on maize, for example, and you want to do cultivar identification, then you have to look within the sequences of all the maize varieties or lines that has been submitted. Now, for example, maize is a crop that is grown in Latin America, all over the places, right? So it will be very difficult for you to, find the line if you are looking for a line in Nigeria, for example. So it's better to then look at, the sequences that are available for Nigerian lines. But sometimes it can happen that, those lines are, the sequences for those Nigerian lines are not available in public domain. So it's just not NCBI, but you also have phytosome. There are different gene banks. So you can then go, if you don't get the Nigerian lines, for example, then you can look at the global lines and see where, if 90% of your target DNA for your target genotype actually matches, then you can do the cultivar identification. Yes.
Thank you, Ranjana. Matsikwan, let us know if that answers your question. There's another question on GWAS on the chat by Diriba. For how many genotypes is it appropriate to conduct GWAS analysis, since different researchers discuss differently?
I think I have answered that question.
Okay. I thought you had answered only the three points before. Apologies. So we'll go to Marta, who has her hand up then. Sorry. Marta, you have the floor.
Okay, thank you so much for the great presentation. My question goes to Zakaria. most of the time in our gene bank, we have the issue of duplication. So farmer normally name the accession in different name. So we end up having in the gene bank different name of variety, but a lot of duplication. So I would like to get from you which insights, how could we manage better our gene bank using genomic tools to avoid the issue of duplication while collecting the samples? Thank you.
Yeah, it's something that we are thinking. I think those tools, and again, we look at the presentation before. Those tools are very, very important to. I think now we are able to have how I call it set of markers. They can be even low density, very low, cheap, that we need to use before actually finalizing the whole process of conserving a sample. Let's say the best thing will be like, let's take a species barley, for example. If what we are trying to do, we are trying to identify set of markers or panel of markers that will help us really differentiate about what's new and what's not new, and identify duplicates. So when we go and collect, it's better to run very, very quickly set of marker, hopefully not expensive, to be able to only put in the gene bank collection as new material, the one that it's unique. That's how it should be. And it doesn't need to be millions of markers. It can be very little sets. Maybe Ranjana has an answer there.
Yeah, no, there's no answer, but this is something that all gene banks, struggle with, Martha. And clonally propagated crops are actually much more worse than the seed crops, right? Because in clonally propagated crops, you are just doing it through, like yams, through sets, right? There is an issue of just not synonymy, but also homonymy, where you have different accessions with the same name. So you don't just have duplicates, but also how same name is given to the different accessions. So how do you manage that? It has a huge implication, just not for maintenance and management, but also in selection of parents for further breeding, because this is part of the pre-breeding, right? And Zach is right. You need to, whenever you go for collection, you need to know, just with the markers, cast markers, which are very cheap, 10 markers can actually, 8 to 10 markers can actually differentiate your materials when you go for collection. But also the morphological descriptors, right? You need to have those distinct, mainly the qualitative traits. For example, pigmentation, anthocyanin pigmentation. Do you have that? I know it could be a bit tricky with the morphological data, but you can take those, kind of the distinctness, sometimes, whether it is like that, and then combine both morphological data or characteristics with your genotyping with 10 markers to distinguish. But this is something which we have been battling for last, I don't know how many years, 20 years maybe, Zach, in the gene banks, right? I remember I did the core collection of pearl millet during my PhD in 2001, and we keep doing mini core collection, these collection, working collection, because we are all the scientists, we are trying to minimize the duplicates, because it has a huge implication in the further breeding programs. Thank you.
Thank you so much, colleagues, for that intervention. So I'm checking. Yeah, I saw that Arnaud sent a response to your answers. So yeah, there's more questions for you, Zach, related to that. I don't know if you want to take that now or offline. We are still waiting for Carolina, but I think she may be having trouble joining. So Zach, do you want to take those two questions? They're in the chat.
Yeah, I think Leah is, Leah and admixture, what we call structure, it's almost the same thing, really. It's different, how I call it, different engine behind, but it's exactly the same theory.
Algorithms.
Yeah, algorithms.
Yeah.
No, actually, yeah. It's exactly the same theory, but the algorithms are different. It's just Leia is a little bit more fast, and if but I feel that Leia is a little bit more complicated to understand than normal admixture we have in structure analysis based in Bayesian. I'm not saying Bayesian is easy, but I feel that yeah, structure, for example, software is much more easier to interpret than Leia in terms of what's happening inside. I hope that answered. The other one is often mentioned. It's going up. I don't. Now I'm going to another one. Yeah. Okay. How close the accession should be to be
No, it
considered.
is about linkage disequilibrium, Zach. It says in
I
GWAS,
don't
it is
see it
often
anymore.
mentioned that linkage disequilibrium plays a very important role in explaining or confirming the identified candidate SNPs. Could you please elaborate on the role of linkage disequilibrium and its usefulness in GWAS? You are muted, Zach.
Hello? Do you hear me now?
Yeah.
Yeah, I think the concept of linkage equilibrium, if I get it, if I may, linkage equilibrium means that the locus of marker is not linked to the variation in another one, correct? Yeah, so that's very clear. So what we are looking in GWAS.
Thanks so much, Zach, for that response. So I still cannot hear you. You are muted. Are you talking?
He's muted again, yeah.
I see his lips moving, but no sound. You did not finish. Yeah, your audio cut off, unfortunately. Yeah. Sorry, Zach. Okay. We can wait for you. In the meantime, I see that Carolina is connected. So Carolina, could you maybe
Alessandra, can I just respond to this question? How close the accessions should be to be considered duplicates? Again, it depends upon the genetic distances that you get. You know, whether do you get a genetic distance between 0 and 0.6 across all the genotypes that you have been teaching, you have been using. For me, I always look at the lowest distance, between 0.05 or 0.1 to consider them as, kind of genetic distance to be considered different, So that's what it is. So the genetic similarity, if it is too high, that around 96, 97, 98 or more, then they are similar. They are more identical. So this all depends upon, how the genetic distances are within your material. So anything beyond 0.95 or 0.9, they should be all considered duplicates, or even beyond 0.8, probably they are duplicates. It also depends upon the number of markers you have used. So this is something that you can see, look at how your data is behaving and how your genetic distances are behaving. Yeah. So try to say that the genetic distance is high, between the two comparing accession. Hope it answered.
Thanks so much, Ranjana, for taking that on. I see we have Carolina connected. I just wanted to make sure that she can hear us, her microphone is working, so we can proceed with the next presentation. And if you have more questions for Ranjana and Zakaria, you can still ask them writing in the chat. And if they have time, they will respond here. Otherwise, we can set it up via email. So Carolina, can you hear us?
Yes, I hope you can also hear me.
Yes, perfect.
Oh, great.
So,
I was having problem with the microphone. I apologize.
oh, sorry, it's okay. I'm glad you made it. Do you want to try and share your presentation if you have one?
Yes.
Go ahead. It's loading. Perfect.
Can you
I can see
see?
clearly.
But
Yes.
can you see in presentation mode?
Yeah, we see the full screen.
Ah, okay, okay. My screen is different. Okay.
The floor is yours.
Thank you.
Thank
Hello,
you.
everyone, and I'm sorry for all these problems. Well, following, I think we will have a lot of relation with Zakaria's presentation, but I will give you a brief a summary of examples of the uses of DSI, specifically for Genebank, but also can be applied for other crops, other approaches like breeding or other type of areas. But the main focus will be in Genebank. Of course, you are more aware that all the challenge we are having to feed the world sustainably. We have a lot of issues with water scarcity, climate change, of course, poor soil and fertility, new diseases that are coming. Also drought heat, the growing population mainly, it's a big concern, and malnutrition and desnutrition. But we have one good thing among all these challenge, which is the genetic resources we hold in the gene bank that in the majority of the cases are still underutilized. If we consider the future of increases in yield, this example is for maize and wheat because I'm from CIMMYT, as you know. The curve going through a normal breeding program is growing. Yes, the breeding are very efficient, but the estimations say that we will not reach that demand only with conventional breeding. So we need to intervene with the genetic resource and with new technologies to change the curve and reach the demand that is expected for 2050. So as you are aware, most of you come from the gene bank. In the gene bank, we hold all the diversity, but out there in the breeding program and in the farmers field, the diversity is reduced through of course, the breeding cycles. No, every time the diversity, while we improve materials, we reduce this diversity. So, in an analysis that we were gene bank, we found that only 6% of the diversity has been used by breeding program, but it still remain 94% with great potential for different things. No, could be potential for climate adaptation, resistance to diseases, nutrition, quality, etc. So there is a lot of opportunities. Sorry. In the gene bank. Again, as I say, many challenges for food security and nutrition. So the breeders need new genetic variation for trait improvement. And the gene bank are the reserve of this variation. So first, we need to understand the collection in order to provide a better accession to breeders and farmers. As an example, in the CIMI gene bank, we conserved 28,000 maize accession and 124,000 wheat accession. It's very large collection. So how we can identify which are the best accession for the specific challenge? Well, as I mentioned, a very large proportion of the gene bank is still underutilized, is still sitting in the gene bank. We need to understand this collection first. It will be great if we can phenotype the entire collection, but depending on the size of the collection, in our case, is super large. So it's not an option to have all this information. It's an activity that we are doing, yes, but not for the whole collection. Genotyping, or DSI, digital sequence information, I'm sorry, is an efficient method with has unbiased and is cost effective.
Sorry, Caro, your audio is breaking off. We cannot really hear you.
Can you hear me now?
Yeah. Now it's better, yes. Not sure what was going on.
I'm coughing a little bit, sorry.
No, don't worry. Go ahead.
So if we compare the genotyping with phenotyping, of course, the genotyping cost is less. It can be done in faster, and we don't have interaction with the environment. While phenotyping, the cost is high, it takes more time because we need to consider the cycle depending on the trait. And of course, we have interaction with the environment depending on where our experiment or our evaluation is. So of course, we have some bias in the phenotyping. So what we did here at the CIMI gene bank, I presented this slide before. We went through a characterization, molecular characterization of the gene bank using, in this case, Darcy technology. So we took a sample of all seeds in the gene bank, we planted in greenhouses, we extracted DNA, and then we genotype. In our case, in-house, in the lab called Saga, but of course, this is a Darcy technology coming from Australia. And this is one of the results that we implement the DSI, all this genotyping information. First, we wanted to answer how the diversity of our collection looks. And this is an example only for the hexaploid wheat. The hexaploid wheat has the A, B, and D genome. In this case, we combine in the same analysis 60,000 accessions. The good thing is not only CIMI material, but also we combine with ICARDA material. So we have opportunity to look at more diverse collection. In this hexaploid wheat, we include 8 domesticated species and coming from over 100 countries, and we include 85,000 SNPs. As you can see, this is quite nice picture. I will start again. In blue, you can see the land races. In red, the breeding material, and in green, the synthetic wheat. Synthetic wheat is a durum wheat cross with a wild relative in some cases, and create synthetic wheat. We create new diversity that is not existing in land races. or elite material. And it's very evident here how the green is bringing a new dimension of diversity. And we also see, for example, here, how the breeding material is in a corner, meaning that the diversity it's reduced. We can see this bottleneck of diversity, and we can also identify areas of opportunities. For example, I don't know if you see my pointer, but in the corner, in the blue corner, you can see that no breeding material is present in this part. So means that there are still opportunities and land races underutilized that need to be explored. Doesn't mean that are all good. If it's not explored, it's good. Not. But we need to give an opportunity and explore what is there and what potential can bring these land races. With the same study, we did a cluster analysis. So basically, we wanted to divide in groups all our collections to understand more in detail what we conserve. So we started by first two big groups, the blue one and the red. The blue one, it's more original material, all material coming from center of origin. The red one is more modern material and include improved varieties as well. So all these blocks here in the middle are the breeding material or elite varieties. And you see that it's not breaking. So again, the diversity is compact, no? So it's not breaking this group until by the end. So this is an exercise that we did, and we started to clusterize and put in some tag to each group. So these are Mexican land races, or then we have the land races from Turkey, land races from China, land races from other countries from the center of origin. So it's very nice how using this technology we can group the diversity. Also, of course, we did a admixture analysis to see the structure of all this group. So we could understand how is the diversity also within each of the identified groups. Sorry. So then we took these first two groups, the red one and the blue one, and we wanted to zoom in inside the genome. So this part, it's a little portion of the chromosome A, and this part is also the same portion of chromosome A from the red. So this is the blue, and this part is the original material in blue and the modern material in red. And on the right, it's just a zooming of this region, because in this region, all these dots are the FST of each SNP, and the red one are the significant SNP. So we've positioned one gene here, the pre-harvest protein gene, in this region, and we zoom in to look more closely the SNP associated with this gene. As you can see, oh, sorry, this move, it was a little bit to the right. So as you can see, there are some red dots. So SNPs here in the original material, but then when we move to the modern material, it disappears. So what that means? Well, it means that through this evolution or breeding, there is definitely a bottleneck of diversity, and some alleles are missing in the current elite varieties. Again, doesn't mean that this is good or bad. It means that on the process of breeding, there are alleles that have been selected, probably looking at yield or looking at some specific trait, but not all traits are considered, no? So this allele could be left behind in the breeding process. Again, it's an evidence that still we have diversity there, underutilized, and we need to look at what are the effect of these alleles, and if they are good effect, can be potential introduced in the breeding programs. But of course, we are talking about exactly the same study we did with exactly this study. We did with the tetraploid wheat and with the wild relative. I'm not presented because it will be long, but we have exactly the same study for these three different groups. But we are talking about 124,000 accession. So, of course, for the breeder, it's still a challenge. So we decided to perform a core set or a subset of this diversity. So we reduce 99% of the collection. We selected only from these 60,000, only 1,500 accession to evaluate in the field. The core set can be different sizes, but this was the size that the breeder have a space in the field for evaluation. So we try to conserve as much as possible the diversity, more than 99% of the diversity, but reducing significantly the size of the collection. So again, all this technology, all these molecular markers help us to reduce the collection at this size without losing diversity. These are two examples also of two core sets that we did. We selected all Iranian land races that we have in the gene bank, a little bit more than 2,000. And we did the same study, the same diversity analysis, and we reduced this collection to 300. Similarly, with the Mexican land races, of course, because we are in Mexico, we have a large collection, more than 8,000 accessions from Mexico. It's not the center of origin, but we still have a lot of diversity that many years ago was introduced by the center of origin. And with it, we did a core set of more than 1,000. But as you can see, the reduction of the size of the collection is evident. So these two core collection has been evaluated in the field for different traits. And now we can make a lot of decisions. Selection, actually, it was selected for breeding material, for pre-breeding programs based on the evaluation in the field. So I'm showing different approaches that can be, that molecular marker or the DSI can be used for gene bank, no? Another approach that probably Zacaria's presented, it's the genome-wide association studies. So it's a combination of molecular markers with phenotypic data evaluated in the field, and then we can identify genomic region controlling these traits in different chromosomes. So this is a good approach to identify or to be close to genes. Sometimes it's hard in complex traits, but in more simple trait, it's fine. But now we are moving to a new approach. We call allele mining. And we use exactly the same data, but instead of including phenotypic information, we include climatic data, which is basically free. So it's not costly like phenotypic data. So by the combination, and we use this climatic data as a normal phenotypic information. So the combination of molecular markers with the environmental data, again, we can plot genomic region controlling this associated with adaptation rather than trait performance. So it's a different approach, but with a similar concept. We did this for maize, and we are doing for wheat as well. In maize, 7 to 8% of the variation associated with the potential adaptation, in this case to drought and heat, are in maize, yes, I'm talking about maize, are unique in land races. And only 12 to 17% are very rare. So it's less than 1% of the whole collection of maize. So with this approach, we managed to identify unique alleles with great potential for climate adaptation. So this is a summary of the allele mining, again coming from the Genebank. The accession bring in most of the cases passport data, and I'm saying most of the cases because the Genebank is not perfect. We have a lot of missing information, not missing at all. Sometimes we know the country of origin, for example, but we don't know the geolocation. So when we know the geolocation, then there is another step which is very important for me, is the curation of the geolocation. No? Sometimes we see a geolocation that is up in the mountain or is in the middle of the ocean. So we need definitely curate all this data before applying a methodology like this. So we've been doing this. We completed the maize collection, and we are almost finishing the wheat collection. Of course, for the data available, and if we don't have available, we are even looking at historical books to see if we can find the collection missions and some information of the town on. So it's very, very tricky and very demanding activity, but very necessary. Otherwise, this study is not good. So having a curated geolocation data from the accession, then we can extract climatic data for the last, I don't know, 20, 30, 50 years. And this is available and is almost free. So again, passport data with geolocation, we know exactly where these accessions were collected. We can extract the climatic data from this specific point of geolocation and perform an environmental GWAS study. But again, this is the first part, how we move to breeding program. So in the Genebank, it's like we conserve land races. It's like conserving a tiger. We don't want a tiger in our houses, right? So we need to go through an improve or breeding program, in this case, a pre-breeding program, because we are not even there. Huh? So of course, we don't want a tiger in our house. So we need to go through a pre-breeding phase, understanding the relationship between the genotype and the phenotype. And in the pre-breeding, we will have like a not very nice cat, but something that looked like at least like a cat. Finally, the breeders will finalize these varieties and produce a very nice cat that we will be happy to have at home. So this is similar what is happening in the breeding program of crops. If we see here, you can easily see the difference between the land races and the lead varieties in wheat. Here is in maize. Unfortunately, now we have in the field super tall hala race in maize, which is up to 5 meters. It's crazy, or even more, 6 meters. I forgot to take picture because it's very nice to see this hala race. But of course, when you see that, and the corn is very long, but it's not good in yield, even if the corn is big. So of course we need to go through it, and then it brings a lot of opportunities. No, it's resistant to some diseases, but if we plant in the field, it fell, and well, I can share some picture with you. Unfortunately, yesterday I wanted to take picture and I couldn't, but it's very nice. So definitely you see the difference between our tigers and our nice cat. So this is what the breeders and pre-breeders do from Genebanks. So again, the DSI can assist in this process to speed this process and to identify more precisely and with more efficiency the land races from the gene bank. Another approach is genomic selection. You know also how it works when we have a training population and we predict the performance from some land races, and we can extract these land races for evaluation and for including in the and to include in the breeding program. So how is the vision? I say that without DSI or without, of course, phenotypic evaluation, the gene bank looked like a supermarket without labels. No? If we go to a supermarket to buy some specific thing and we don't have labels, how we start? Which one has the bin that we want? We need to start opening can by can or envelope by envelope and look what is there and check. So it's an activity that will be forever. So by using genomic approach, molecular markers, we can put this label into the accession. We can identify not only the passport data that bring country of origin, geolocation, and few others data, but not much. We can add value to this accession by saying, okay, this corn that if we have two similar corn, we can say, okay, this is good for popcorn. And this also is good for popcorn, but not both of them are good for heat tolerant, for example. So we can tag this information into the passport data, and so the breeders can come and search more efficiently and faster. Well, and this was a brief summary of the approaches we use, some of the approaches. Actually, some slide is missing. I don't know what happened, but I also included other approaches that we are doing. It's checking the redundancy of the collection. We didn't finalize, we have a preliminary analysis, but because we didn't complete the collection, now we are completing the genotyping of the collection. It was not fair to mention if there is a big redundancy or not, because we don't have all the accession genotype. Now we have, so we will redo the analysis with all land races and see what is the level of duplication. Also, we want to compare with other gene bank conserving wheat, like for example, Icarda. So what is the level of redundancy? What are the unique material that we conserve, and what are the unique material that Icarda conserve? We can create a core set across gene banks. I mean, the opportunities are very big. We also We also did, I don't know what happened with my slide, but there are two missing now. The curation of the taxonomy. This is another very important thing that we managed to do with the molecular information. In this case, we found that in the collection, we had a mix accession in the passport data saying that it's durum wheat, and we found a lot of molecular markers in the D genome. So durum wheat is a tetraploid, have A and B genome. But we found 1/3 of the markers in the D genome. So of course, this is a mislabel. It's not a tetraploid wheat, it's an hexaploid wheat, and vice versa. So with this data, we could fix the passport, and of course, not just by looking at the data, but we took this accession out. We look, but before that, we never look at the accession without knowing that the molecular markers say this is not a tetraploid, this is an hexaploid, or this is not an hexaploid, this is a tetraploid. So these are nice things that we can do, especially for large collection, in a faster way. And now talking more of how we presented the first day, the second day, sorry, that we are working in a Latin American community of practice using molecular markers. So I'm going to present one experience, one example of one collaborator from Embrapa in Brazil, to show you the strengths of this community of practice, and something that will be really nice to replicate in other regions as well. So in this case, I'm presenting for a group that work with beans. In this community, all the national gene bank from Latin America presented what crops they are working, and we divided in they generated data with us. First, they analyzed the data by gene bank, but after we combined the same crop from different gene banks. So it went beyond the unique gene bank. And this is, I think, one of the strengths of the community, because we look for opportunities outside. It's not only my gene bank. I'm comparing my gene bank with the rest of gene bank in the region. So in this case, I'm presenting the bean group, and we have people from Brazil, Embrapa, from Argentina, in INTA, from Chile, INEA, other INEA from Peru, Colombia, and Mexico. So it's different countries. As you can see here, all the countries, the institutions, and the accession they provided. Of course, it's not large collections, they are a small collection, and are the accession they could provide for DNA extraction, and we started to understand what we conserve. It's not perfect, yes, but look at the number of SNP, how by individual collection, we have 10,000, 11,000, 9,000, 8,000, but by combining all, we increase to up to 19,000. So again, we are increasing the number of markers. Why? Because we are increasing the diversity from the accessions. And this is one preliminary study that they performed. In Latin America, we have 2 gene pools. Bean is common bean, and were domesticated from 2 centers of origin, the Andes and the Mesoamerica. And you can see very clearly the result of combining all these accession from different gene banks, how is differentiated very nicely the 2 center of origin, the Andeans and the Mesoamerican. And then, of course, they are evaluating inside each group, answering a lot of questions. So there are a lot of potential in this study. Similar analysis, it's plot in 2 dimensions in the PCA. And the same study that we did in wheat, they did here in beans by clustering this collection in subsets. And these are I wanted to share with you because are quite nice, are comments coming from them. So how in this case, our collaborator in Embrapa benefit from participating in this community of practice. And I think it's nice to share because if we want to replicate, you can see what are the benefits that they are having by right now. It's the 4th year of the community, no? So it's not just one course and that's it. We are together for 4 years already, working together, interacting, exchanging ideas, exchanging material, making scientific publications together, exchanging SOPs, for example. So it is a community of practice. It's more beyond that course, no? So how Embrapa in this case benefit from the community? They say counting the genetic diversity held in our gene bank compared with other gene banks, training on modern tools and methods, and facilitate access to high throughput molecular analysis at a good cost. Regional networking, improving the chance of participating in collaborative projects and international funding. And the benefit now for the BEAN group. I mentioned all the countries involved in this group. Well, they could better understand the bean germoplants in the continent by exchanging passport data, phenotypic information, and also genotypic data. Exploring a broader allele diversity by pooling data on germplants from very diverse environment. And now they want to do more ambitious perspective for future. They want to create a continental core collection of bean, sparse testing and genomic prediction, germplant stem exchange to mitigate climatic challenge. So they are looking into the future by themselves and looking for opportunities to continue collaborating as a group. And well, the potential benefit for the whole community is, of course, the networking of the Latin American national partners with CGIAR centers and some companies as well that are involved, promoting downstream adaptation of the different technologies that we presented here in crop genetic resources. Continental capacity to conduct survey and diagnostic and articulate competitive response to funding opportunities. Now we are applying also for some funds as a community, not as a CIMID or CIAR or National Gene Bank. We are approaching donors and looking for some opportunities as a group of Latin American national gene banks. And a pipeline to promote the flow and use of crop genetic resources under, of course, the principle of FAO and treaty. So with that, I would like to thank you for your attention, and I hope this gives you an idea of some of the uses that us as a gene bank from CG use with molecular markers, genomic technology, and in the end, DSI. And some examples at the regional level, how our collaborator, our partner use this technology, this data, and this knowledge for their own national gene banks and in collaboration with other partners as well in the other countries. So I hope it clear, and I'm happy to answer any questions.
Thank you, Carolina. You got a lot of applause. I don't know if you could see that.
Thank
So
you. I can
I
see.
think- I think it was a successful presentation on that front. So the floor is open for questions. Okay, I see there's a hand up, so I'm going to give Elizabeth the word. Elizabeth, you have the floor.
Thank you very much. Where I'm at, it's morning. So good morning, good afternoon, colleagues, to all of you. Fantastic presentation, Carolina. And also, you know, I want to thank Zach for actually clearing up a lot of myths before about the software, the use of the software, especially structure for me personally. But Carolina, I also wanted to ask on the question as to cost. How much does this actually cost to do? It's great to see the work and of course you're building with wheat and corn, you're building on a foundation of, you know, nearly a century of data, right? Information that was there before. When we talk about tropical crops, you know, that's not there. We're still starting. So some guidance on, you know, how not to fall into pitfalls, you know, what were the lessons learned? What are the key things to start collecting information, to start collecting on our tropical crops, and to help us get to the stage where we can start, you know, using these tools efficiently, both cost and time efficient, because especially in countries like the Caribbean, where you have an entire island with just about 100,000 people, you could imagine, you know, you don't have a lot of labor force and capacity. So, you know, what are some of those gems you can give us on how to approach getting and managing the knowledge of our genetic resources effectively? Thank you so much.
Thank you, and it's a great question. I think first, you are not alone. I think this is very important. You have the support of a community again, so you don't need to start from zero. And as you mentioned, very good. We have the foundation, we have the experience. Not only us, different gene banks. Zakaria has a lot of experience in his gene bank. So we have a team that can support in these activities, so you don't replicate the errors that we did at the beginning. That are few, to be honest. Just to give you some examples. At the beginning of this project, of the genotyping project, it took over 10 years, and it was very large project funded by the Mexican government. When we started, we didn't have any platform, for example, for tracking samples. So it was done all in Excel, and we were, you know, I was not in this phase, but anyway, the people were checking and registering in Excel, giving a name that is 1, 2, 3, 4, 5. At the end, we have 100 of ones, 100 of two. You know, it was a lack of sample tracking. So then a sample tracker system was developed. It's also open, and there are more than one today that can be used. So to start with, first, make really sure that you track properly the samples, because generation of data is easy. But then going back to the original accession, if you don't have good tracking, it could be a big problem. And at the end, is wasting of resources, time, people, because you invest a lot, and then at the end, the results you get, you cannot go back to your accession. So sample tracking is one of the, look like very obvious, but not all are working on this area. Are more focusing in generation of data, quickly genotypic information, but sometimes they forget about tracking properly the accession flow, no? All the steps. So this is first. Then the cost. We did in our case in-house because we have the technology here. We can also offer this technology to anyone that want to access. Similar technology is provided in Australia as well. The difference is, the technology is exactly the same. The difference is, as we are a CG center, and we are not just a service provider. We want to also collaborate, interact with the partners, do something together. This is more the approach. It's not business, it's more collaboration, no? So the cost could vary depending of how diverse is the germ plants. We have different levels of, and I say we, but I'm from the Genebank. I'm not from the genotyping platform anymore. I've been there for many years, but now I'm managing the Genebank. So there are different densities. High density, medium density, low density. So again, it's very important to understand the objective of the genotyping. What is the purpose? Is to understand the diversity, the complexity of the genome. So there's different questions that we need to look to say, okay, maybe high density for wheat, for example, we use high density, which today it costs $40, $45 per sample. In some cases, with mid density is enough, which costs $30, $35. And the range is because of the number of samples. If you send a lot of samples, the cost reduce. And for other approaches, low density is more than enough, which is $20, $25. So it depends of the purpose, of the complexity of the crop, like a combination, right? But it varies between $20 to $40 per sample. And all the work that we did for the community of practice, we supported. They didn't pay anything. We supported with the funds come from the Genebank Accelerator. And so it was, we selected people from the community, and they sent 200 samples to us free of charge. We generated data, but of course, the idea was to use this data as a team. No, they can use also for their own purpose, for their own understanding of the Genebank, but they need to provide also the data to work as a team. And it was free. So yes, again, to summarize, you are not alone. Please reach out and tell us what you need, and we can support without any problem. And tell more detail of our experience. Sorry, I was a bit long.
No, thank you very much. You answered my questions. Thank you.
Let me open the chat.
Yeah, I'm monitoring as well. Thank you so much. You see, there's a question from Arnaud. First, there is a question from Zach. I don't know if you've already answered that in your very extensive response now. Otherwise, there's a question from Arnaud. Can you see them in the chat?
I see a lot of questions. Can you read, because I'm a bit lost?
Yes, no worries. So Zach said, thanks, Carolina, for making the point on samples tracking. Most of the issues I found while analyzing data for GS and GWAS were linked to false matching in IDs between genotypes and phenotypes. This is more of a statement.
Yes, thank you, Zach. Yeah.
Then Arnaud asked, obviously, thank you very much for the excellent presentation. Let's assume that after conducting a diversity analysis of my collection, I identify a group of 50 accessions, of which 45 come from Africa and only 5 are reported as coming from Europe. Based on the passport data I have, can I assume that these 5 accessions actually originated from Africa and that their previously reported origin data may have been incorrect?
Not necessary, I will say. I don't know, Zach, please come as well, but I think it's not necessary. We have, I mean, this is a different approach. No, you can do a, how it called, the evolution path. No, it's different approach to see if come from the same origin. No, but if the environment maybe are similar, not necessarily are coming all from Africa. No, and we need to really look at this. We did, for example, for maize, the evolution path, and it's nice to see how from Mexico it was spread to other countries. No, so yeah, we need to. I will not assume. It's a possibility. Yes, it is a possibility, and we found a lot of mistake in passport data. Imagine that we have accession 80 years old. So of course, 80 years ago, we didn't have the technology. We didn't have GPS. We didn't. So that's why we are looking into historical books to see, reading the expedition missions, and so it's very complex. But I would not say yes. It needs more analysis. But very interesting. Very interesting.
Thanks so much, Caro, for that response. There was a comment about whether people will receive the recorded presentation. Yes, absolutely. All presentations will be shared, as well as the recordings at the end of the webinar series. So that's on September 12th, when we'll have our last session. I think, Caro, if you could stop sharing. I don't see any hands up. So, and we are also out of time. So I think we are good for today. Marie-Noelle, I leave you the floor to close today's meeting if you have anything to add.
Yes, thank you. Thank you so much, Alexandra. Thank you, Carolina, for the nice presentation. Thank you for Ranjana. Thank you for Zach for the nice presentation. Please join me in uploading the 3 excellent presenters we had today. So tomorrow we will continue with the presentation on the use of GSI in arid crop conserve at ICRISAT. So as well as the study of genetic diversity, the development of core collection. So kindly connect on time tomorrow. We will start at 1 p.m. European time. And then for the 2 presentations we scheduled for tomorrow. So thank you all and see you tomorrow. Thank you.
Thank you. Bye-bye.
Bye-bye.